the team from the Ontario Institute for Cancer Research, Mark Mabanglo and Mahmoud Noureldin. So Mark got his PhD in chemistry from the University of Utah, where he trained in mechanistic enzymology, and then he did a structural biology postdoc at Duke in the lab of Lorena Bies, and is now a research scientist at the OICR. Mahmoud did his PhD in pharmacology and physiology at the Western University in London, Ontario, and then joined the University of Toronto before currently being a postdoc at the OICR as well. And today they will tell us all about DCAF1 as a protac hijackable ligase, sharing some structures and insights into substrate recognition. So the floor is yours. We're very excited for your presentation.
All right, thank you so much. Okay, can you see my slides?
Yes, that looks great.
All right, perfect. Let me get my pointer. Okay, so hello everyone. Thank you for joining us today. My name is Mahmoud, and today I'll be presenting some background on the DCAF1 protac project, the discovery of the early DCAF1 ligand, and the biology behind our recent paper. My co-author Mark will cover the structural part later in the talk. So first, I would like to briefly introduce our institute from Canada, Ontario. The Ontario Institute for Cancer Research is one of the leading institutes in Canada. On the left, we can see just the blueprint of the institute with three key themes. We, the drug discovery team, belong to the therapeutic innovation arm. And then within our team, we are divided into four sub teams: chemistry, structural biology, biophysical screening, and discovery biology. Within our program, we have like proprietary discovery programs and open science programs where we collaborate with scientists from the community, or we would have our own in-house initiatives, such as the WD-40 platform. And within this platform and with our collaborators, the SGC, the DCAF1 project was initiated. So, as everyone knows, protacs are bifunctional molecules, harness the ubiquitin proteasome system to target proteins. It's a rapidly advancing field with over 30 protacs currently in the clinics. As everyone know, most protacs rely either on cereblon or VHL, and that is why exploring underexplored E3 ligases such as DCAF1 is important to help us navigate and tackle emerging resistance mechanisms, for example, or to broaden our targeted protein degradation coverage to more cell lines, tissues, and different subcellular compartments. So DCAF1, also known as VPR binding protein, was discovered due to its association with the HIV accessory protein called VPR, hence the name. It belongs to the WD-40 repeat protein family, which is characterized by the presence of a beta propeller domain, and they facilitate protein-protein interactions. DCAF1 is part of two E3 ligase, the colon ring ligase and the EDVP, where it functions as the substrate receptors to facilitate the ubiquitination of the substrate, resulting in obviously the degradation, but also affects localization, interaction, and activity of the proteins. Here is the summary of DCAF, some of its functions, and it functions primarily through its E3 ligase. It regulates multiple pathways by controlling a diverse range of substrates, and it's been linked to tumor development in the literature. So before we discuss the DCAF1 WDR5 protac, I want to go very quickly and briefly over our JMED paper published in 2023. A collaboration between XChem, ZBI, now acquired by Relay, and OICR discovered the first nanomolar ligand against the WD-40 domain of DCAF1. And this was the starting point for our DCAF1 protac program. So a DNA-encoded library and a machine learning identified 490 nanomolar hit or ligand against the WD-40 domain of DCAF1. DCAF1 is a very big protein, and the screen was done on only the WD. This hit was confirmed by SPR, and then our collaborator at the SGC were able to successfully co-crystallize, and we we found that the ligand binds to the top pocket of the DCAF1 domain. This was very important as it allowed our medicinal or computational chemists at OICR to start SAR design and optimize a more potent compound. And then we reached 38268, which was around 35 nanomolar. And this was our best compound at the time, able to crystallize that. And then here just summarize the biophysical characterization of this new compound by SPR. Like I said, was around 35 nanomolar. And this compound was able to thermally stabilize the WD domain of DCAF1 in vitro by DSF. And then after that, I developed a JES and hybrid-based SETSA to confirm target engagement of this compound in cells. Starting by the JES-based SETSA, as we see here, 26E or 38268 was able to thermally stabilize the WD domain in cells, as we see here across later temperatures. And also this was dose-dependent. And then also the hybrid SETSA basically correlates the results. We're able to thermally stabilize across different temperatures. And we had around 10 micromolar EC50 at the time. And obviously, we wanted to optimize our ligand to achieve better in-cell target engagement for our DCAF1 protac development. And while we were working on that, two DCAF1 recruiting protacs were developed. One, they used an irreversible covalent binder against DCAF1, and then Novartis developed DCAF1 to degrade BRD9 and BTK. So after optimizing our ligand, we moved forward to develop and structurally characterize our DCAF1 WDR5 protacs. And this resulted in this study, which focused on using DCAF1 to degrade WDR5, a well-validated cancer target involved in gene regulation and histone modification. So the aim was to explore DCAF1, a less commonly used E3 ligase receptor, to use as a protac. Here we have the crystal structure of DCAF1 with the 38268 compound that I mentioned, the JMAD paper. And then we did a perazole to a pearl switch, which helped us to kind of extend up toward the surface and to develop the protacs. Here's the chemical structure of the WDR5 binder. This binder was developed by OICR here, 2017. And then here's the linkers used to create the bifunctional protac molecules. So I'm showing you here the first generation DCAF1 WDR5 protacs. On the left here, you will see the SPR KD against WDR5, and here against DCAF1. And from the get-go, you see here as we increase the linker length, here was a 6 pegged versus 4, we start to see better degradation and potency by degrading the hybrid WDR5, the exogenous hybrid WDR5, and the endogenous. So this result confirmed that linker optimization can enhance protac performance, and then we went through various range of linkers. And then we reached our more optimized linker length, protac number 4, which was a 9-peg linker. And this resulted in a much greater Dmax and an improved potency compared to the other shorter linkers. And I benchmarked our protac against VHL-based WDR5, and we were pretty comparable in terms of the Dmax. So for my last slides, I'm just going to show you this slide summarizing in-cell characterization of protac 4. Here we're just confirming that the protac can degrade the endogenous WDR5. And then this degradation was prednisone dependent. As I titrate MG132, we see here we can rescue the degradation of WDR5. And then lastly, the WDR5 degradation resulted in growth suppression of MV411 after 5 days compound treatment.
Thanks, Mahmoud. Once again, my name is Mark. I'm the X-ray crystallographer of OICR, and I'm part of the protein production and structural biology group of OICR. Sometimes we also do biophysical experiments, and what I'm going to show you now are some of the biophysical characterization of the four protacs that Mahmoud discussed, as well as the X-ray crystallography, which was my primary job. So Mahmoud discussed the first three protacs whose linker lengths range from PEG 4 to PEG 6. And we employed differential scanning fluorimetry to test whether these protacs can bind their targets. And as you can see here, that the protacs 1 to 3 can stabilize the target WDR5 alone or DCAF1 alone. And when the two proteins are mixed together in solution, we can see that there is a shift in the melting profile for the mixture in the presence of the protac, as you can see here. And there's a little bit of shift to the right. So with this DSF method, we thought that it wasn't a cut and dried way to demonstrate that the ternary complex was formed. And so we used another method, surface plasma resonance, to demonstrate that not only did the protacs 1 to 3 bind WDR5 and DCAF1, but also that the ternary complex is being formed. Mohammed and Alice Lee from our group performed the surface plasma resonance for these two protacs. And as you can see from the sensorgrams, that there is a decrease in the dissociation constants for the protacs 1 to 3 in the presence of WDR5, as well as the residence time is reasonable for these protacs. And so based on these numbers, we can tell that not only does the protacs bind DCAF1, which is immobilized in the chip, but the binding is also improved when WDR5 is present. So there's some level of cooperativity in the binding process. So I go now to the main results for the crystallography, which is the central part of the paper that we published in Nature Communications. As we expected, the DCAF1 warheads bound DCAF1 and WDR5 as expected. You can see here. These X-ray structures, the data for these were collected at the Canadian Light Source. The resolution are pretty high, ranging from 1.9 to 2.0 angstroms. And the electron densities for the protacs are very strong, that we were able to account basically for most, if not all, of the atoms for the protacs. So looking at the ternary structures, you can see here that even though the linker lengths for protacs 1 to 3 are different, so this is PEG 4, this is also PEG 4, but slightly longer, and then protac 3 is a PEG 6 linked protac. You can see that the linkers are compressed in the ternary complex to an effective length of 38 to 39 angstroms. And you can see that the WDR5 position for protac 1 changes when you go to protac 2 and protac 3 ternary complex. And this is really because upon compression of the protac, the WDR5 anchor rotates. So it repositions the WDR5 with respect to the DCAF1 here. And with that, there's also a change in the type of interactions that occur at the interface between DCAF1 and WDR5. So for the protac 1 ternary complex, it's mainly electrostatic interactions. And then as you go to protac 2 and 3, there's less electrostatic interactions, and then mainly hydrophobic interactions with protac 3. And then looking closely at the interactions with the linker, you can see that it's mainly water mediated. There's a lot of water molecules present at the interface, and these water molecules interact with the oxygen atoms of the linker for stabilization. A key finding from these structures also is these what we call now the specificity loop, which adjusts its conformation in order to bind or recognize the linker of the protac. So you can see here in the protac 1 ternary complex, the specificity loop looks disengaged. And then with the protac 2, it becomes engaged. So it's sort of hugging this linker. And then with protac 3, because it's a slightly longer linker that's bulging at the interface, we don't really see electron density for much of this specificity loop. So it's rather disordered. And this one is a detailed look at the way the 3 protacs and the ternary complex bind. So there's a rotation of the WDR5 anchor, and then the linkers also have different curvatures or different registers, different bulges. And with that, the specificity loop of DCAF1 rearranges or changes its conformation to recognize or bind this linker. And then here just shows how the orientation of WDR5 changes as you go from protac 1 to 3. So if you place a dummy atom at the center of mass of DCAF1 and WDR5, we showed that there's a shift. There's a change in the tilt by 8 degrees and then a rotation by 152 degrees in going from the protac 1 to the protac 2 and 3 ternary complex. So summarizing the first 3 protacs, we can see that in going from PEG4 to PEG6, the linker shape changes, and then the specificity loops also respond to the shape of these linkers. Now, looking at these structures, shown here on the top is the Dmax values for the degradation of exogenous WDR5. We hypothesized that perhaps if we increase the linker length, we would be able to exceed this degradation efficiency. And so, as Mahmoud mentioned, we created protac 4, which has a PEG9 linker, so it's a longer protac than protac 6. And we used SPR to demonstrate that the ternary complex can be formed using this protac. And then Mahmoud showed these degradation assay earlier. So the Dmax now is greater at 80%, and it's comparable to this VHL-based WDR5 degrader MS67. So I went on to solve the structure of the protac 4 ternary complex. And like the previous ternary structures, I found that the protac 4 is also compressed to the same effective length of about 39 angstroms. And you can see here the strong electron density for the protacs as well. And what's interesting about this structure is that now we see the full formation of these specificity loop. So in previous structures, this specificity loop appears just as a loop or disordered. But here with the protac 4 ternary complex, you can see that this one is fully ordered. And interestingly, the protac 4 linker is now coiled in this way before it goes down to the DCAF1. So what's interesting about this structure is that the ternary complex is able to accommodate this long linker by coiling this portion of protac 4 because there is this extra space in the WDR5 pocket. And you can see this specificity loop not only interacting with this coil, but sort of pushing it to result in this more stable ternary complex. And here, superposition of the 4 protacs, you can see that protac 4 still has the same orientation as the WDR5 warheads of protacs 2 and 3. But because it's a longer linker, it now coils and then turns and then goes back to this decaf-1 anchor. And then when we place this ternary complex, superimpose it to the published structure of the DDB1, decaf-1, VPR, UNG2 complex, we can see that the protac-4 occupies roughly 50% of the volume of the VPR. So we thought then that perhaps if we further increase the length of the linker, the protac would be able to fill more of the space occupied by VPR and perhaps point WDR5 towards the position of UNG2, whereby causing the more effective protacs. Now, placing the ternary complex of protac-4 in the context of the structure of the whole E3 ligase, you can see how decaf-1 using protac-4 positions EDR5 for ubiquitination by E2. I highlight here in this picture in yellow the potential ubiquitination sites on EDR5. So perhaps we can also use this protac-4 ternary complex structure to optimize it so that we can better optimize the ubiquitinability of the EDR5 target. And so summarizing all four protac structures, ternary complex structures, we can see that in going from protac-1 to protac-4, increasing linker lengths leads to more stable ternary complexes, and therefore leading also to an increase in the Dmax for the degradation of exogenous EDR5. And you can see here below the quality for the electron density for the 4 protacs that were solved in the ternary complex. So the key takeaway from my portion of the talk is basically the presence of the specificity loop of decaf-1, which acts like the fingers of a hand that can receive any types of substrates. And perhaps we think that the specificity loop of decaf-1 is also is used by decaf-1 to recognize the different substrates that the EDVP and the CRL4E3 ligase receives. And further supporting the importance of that specificity loop is the structure of the protac-5 binary complex that we solved. So protac-5 is the shortest protac that we worked on. So this is the structure. Even though from SPR, we can see that it can assemble the ternary complex, we cannot solve a full ternary complex with this protac. Rather, what we have is a binary complex where now we see a different phase of EDR5 interacting with decaf-1, and then the protac is absent. We think that this binary complex is mediated by protac-5, because we cannot grow any crystal when we just mix EDR5 and decaf-1 without the protac. And by looking at this structure, we can see this specificity loop recognizing a different phase of EDR5. So this further strengthens what we think this decaf-1 specificity loop does, which is the recognition of different substrates, and in this case, different protac EDR5 pairs. So that's the end of our presentation. Mahmoud and I would be happy to take your questions.